Welcome to this particular lecture on microbial hormone. In this particular lecture, I shall be discussing the different aspects of hormone produced by different microbes like bacteria, cyanobacteria, etc. Actually, the growth hormone produced by plant are commonly known as phytohormone, and they mainly help in the growth and development of plant body. But when we see in case of microbes, we find that there may be different hormone which is similar to phytohormone which are also produced by microbes. So to initiate, I will take up a unique case study where I shall be discussing the infection of a plant body which belongs to Solanaceae family and commonly represented by potato plant by a bacteria called Agrobacterium tumefaciens. Now this particular plant cell is invaded by different rod-shaped bacteria of the Agrobacterium organism which modifies the plant cell, and it will produce a tumorigenic growth commonly in the form of crown gall tumor. And this modification is mainly caused by a unique Ti plasmid which is entering into the cell and modifying the rate of cell division. But we also find that in this particular tumorigenic tissue, there is a unique hormone which is similar to the phytohormone cytokinin. So cytokinin production in this particular plant body has increased and has resulted in the tumorigenic outgrowth. So in other words, the phytohormones are almost similar to microbial hormones, and to some extent they have similar function. So phytohormones can be of different types like auxin, like cytokinin, like ethylene, jasmonic acid, brassinosteroid, gibberellin, abscisic acid. Basically, majority of these hormones are positive hormones that is helping in growth and development, but sometimes they may be inhibitory hormones like abscisic acid which is inhibiting growth or sometimes interacting with the growth promoting substances. Now, phytohormone can be of different types as I have already told you, but when there is a particular bacteria like the rhizobacteria as seen in this diagram, the green bacterial cells invading the plant cell will modify different functions. Like if it is a rhizobacteria, it will promote the growth promoting substances, it will result in the production of antibiotics, it will promote the production of phytohormone, it will help in nitrogen fixation, it will promote solubilization and adsorption of different nutrients, it will help in solubilization of phosphate, and also it will result in iron absorption via siderophore. So in other words, it is because of the rhizobacteria which is invading the plant cell could do so many activities. Now coming to the first major group of hormone that is being produced by bacteria, which is auxin. Now auxin production can be in two ways. It can be tryptophan dependent, it can be tryptophan independent. Now tryptophan is an essential amino acid which will be converted to indole compounds, and these indole compounds finally will be producing indole acetic acid which is a major auxin. But at the same time, it may also be produced from other part like the amino-oxy isobutyric acid will be converted to a series of intermediate, and ultimately it will also produce the indole backbone which finally will be converted to indole acetic acid. So the first part of this process we call it the tryptophan dependent process, and in the second part of the process is called tryptophan independent process. So auxin production in plants and in microbes can be tryptophan dependent or it can be tryptophan independent. Now in case of the comparison of auxin production, we can see that this tryptophan is converted to indole 3-acetyl dioxime which is converted to indole 3-acetonitrile, and finally indole acetic acid is produced. But in case of microbes, it is the tryptophan converted to indole 3-acetamide and finally directly converted to IAA. So what we learn from this, that the indole production in case of microbe is rapid and is not involving many important intermediates. Now, how this indole production can be assayed or bioassay of auxin production? For that, we require a typical Avena coleoptile. Now, this is a typical Avena coleoptile where we know that the shoot tip is producing auxin. So, when the shoot tip is removed, that typical growth is lost or the apical growth is lost. And if this particular shoot tip is placed on an agar block and this agar block is replacing the shoot tip, then we will also see curvature test. So what we can understand that this curvature or shoot tip curvature is brought about by the auxin which is produced by the shoot tip of this Avena coleoptile. Now coming to the next important hormone produced by plant body and also by microbes is the crown gall tumor induced cytokinin production, which is producing this tumorigenic growth in potato plant, in the tip of the potato plant. And this cytokinin biosynthesis starts from the simple adenosine triphosphate, and this adenosine triphosphate will be converted to trans-zeatin riboside, which is finally converted to trans-zeatin, and you know zeatin is actually the ideal cytokinin molecule. And how can we assay this cytokinin? For which we require the xanthium seed. This is the xanthium seed, and this xanthium seed will be cut into small fragments, and these are the actual embryo fragments which will grow in the third day, where we will see there is increase in cytokinin level. So the cytokinin level actually is bringing about the growth of the xanthium embryo. So xanthium seed test is an ideal test for cytokinin bioassay. Now we come to the next important hormone, which is the mevalonate pathway generating the steroid. And this particular molecule is produced from simple intermediates like acetyl coenzyme A, which is converted to mevalonic acid, and then finally it is producing dimethyl allyl pyrophosphate and then to farnesyl pyrophosphate. Now this farnesyl pyrophosphate is a very important intermediate which will produce a series of compounds in microbes, in plants, in animals. And one of the important compound is sterol. And this sterol production, as you know, in animal, it will inhibit the immunogenic reaction, and in case of plant, it will produce different other intermediates, and in case of microbes also, it will produce other secondary metabolites helping in their growth. Now, there is an important sterol which is the lanosterol, and lanosterol production is taken up both by plant and microbes, where there is combination of dimethyl allyl pyrophosphate and isopentenyl pyrophosphate, which is condensing together to produce squalene, and squalene will be cyclicized to produce lanosterol. And this lanosterol is a typical plant steroid and also initiated by the microbial infection. Now coming to some important bacterial hormone and their function. Now in case of a bacteria like Pseudomonas aeruginosa or Bacillus subtilis, different strain, and also in Azospirillum brasiliensis, there is the production of zeatin, and this zeatin production is actually initiated by ZOG or zeatino-glucoside, which is converted to zeatin riboside, finally to trans-zeatin, and then trans-zeatin is converted to a more stable product which is cis-zeatin. And these products or these intermediates can easily be separated by thin layer chromatography method. And this particular initiation of zeatin production will increase the divisional rate in this bacillus or in case of other bacteria as indicated by the cytokinin production. So increased production of cytokinin will initiate more rapid division of the bacteria. This particular colony, a beautiful design, is actually that of Pseudomonas, and the Pseudomonas will grow rapidly when there is auxin production at a higher level. So auxin is promoting the growth of Pseudomonas, and sometimes, as I told you, this production of auxin may be tryptophan dependent, or the growth of the bacteria may be tryptophan or IAA dependent, or sometimes it is IA independent. here the IAA concentration or indole acetic acid concentration is marked by the red portion of the graph, and the blue is denoting the tryptophan concentration in Pseudomonas, and the green is in Bacillus. So what we see is, with the increase in IAA, there is more tryptophan, or in other words, the growth of the bacteria is determined by tryptophan, which is converted to IAA. So IAA production is tryptophan dependent in case of this particular Pseudomonas, and in case of Bacillus it is not fully dependent, which is of course shown by this next slide, where we can see clearly that IAA production is actually independent of tryptophan, because there is no tryptophan in the medium, but even then there is increased IAA production by the different strains like AM3, AM4, AM5, AM6 and AM7 of Azospirillum bacteria. So it indicates that certain bacteria can actually produce auxin or indole acetic acid even in absence of tryptophan. Now, there is another important activity in case of bacteria, which is quorum sensing. And quorum sensing actually is having different stages. The signaling molecule is produced, the signaling molecule comes out, or the signaling molecules is binding with the receptor, and there is activation of specific promoter region, and the gene expression in the bacteria will bring about bacterial attachment, motility, formation of biofilm. And this type of activity is commonly associated with the bacteria present in the teeth etching in human. And this quorum sensing helps them to colonize in an environment which is apparently not friendly to the bacteria. So this type of quorum sensing bi-hormone-like molecule is observed by certain gram-negative bacteria like Vibrio fischeri and E. coli. In the first case, there is the activation of the Vibrio fischeri Lux system by a unique auto-inducer molecule, which is represented by this structure, which actually is acyl homoserine lactone, which activates the Lux operon, and it will result in the typical quorum sensing activity. Whereas in case of E. coli, it is the AI2 or the auto-inducer molecule which is activating the Lux S and the Lux R, and ultimately it is the unique ISR operon is activated, resulting in quorum sensing or colonization in different hosts. Now coming to a unique group of organisms which is not bacteria but very close to bacteria, represented by these grey colonies of Actinomycetes or Streptomyces. In Streptomyces cellicolor and in other species of Streptomyces, there is a unique hormone-like molecule represented by butyrolactone molecules actually activate the production of different secondary metabolite in the Streptomyces griseus, in the Streptomyces virginia, in Streptomyces cellicolor and Streptomyces lavanduli. So what does it mean? It means that this secondary metabolite is produced under the influence of a growth hormone-like compound represented by butyrolactone. Now, definitely this is followed by a unique mechanism of pathogenicity, mechanism of development of sepsis in human or other host in bacteria. Actually, it has been seen that whenever there is a sex hormone present in male or female, the bacterial reaction varies. Like here, in case of the septicemia or sepsis regulation by a hormone-like substance. And this hormone-like substance is nothing but the catecholamines and other steroid hormones present in man. So what happens is, in male, where the steroid hormone production is more, there will be elevation of catecholamines, elevation of cyclic AMP, which will ultimately lead to lipolysis, lipotoxicity and organ damage, and that will ultimately mark the activation of hypersensitive reaction leading to sepsis. So sepsis indication is more in those persons where the steroid hormone production is more. This group of bacteria, the Microbacterium marinum, represented by this orange culture, they actually invade the lung tissue, causing the dreadful disease of tuberculosis. But in rat, it has been seen that the lung lesion has been restricted in the female rat lung because of the production of female sex hormone, the estrogen. And as because it is lacking in male rat, the infection in the lung is more devastating. Coming to another very interesting point, where the Helicobacter, this group of bacteria, is colonizing in the gastric mucosa, causing the dreadful disease of gastric ulcer. In case of female rat, you can see the bacterial colonization is restricted, again because of the female sex hormone. But in absence of the female sex hormone, there will be more development, more colonization of Helicobacter and more incidence of gastric ulcer disease. Similarly, the Coxiella, which is causing the dreadful Q fever, in this case, the female rat is showing restricted infection or reduced Coxiella load in the ovary of female rat. But in case of ovary ectomized rat, there will be more Coxiella infection damaging the spleen, which actually is a very important gland or very important organ in mammal, where the RBC are destroyed and naturally the bacterial load increases in absence of ovary. But sometimes it has been seen that the bacterial infection can increase in presence of female sex hormone. Here what we see is, with the increase in the level of estradiol, which is the female sex hormone, the lung lesion increases and it is reduced with the reduction of estradiol level. So what does it indicate? It indicates if the estradiol level is higher, then naturally that female rat will suffer from more Pseudomonas infection in the lung. There may be the dreadful disease of Salmonella typhi, which is a gram-negative bacteria, and commonly it has been seen that the Salmonella load in the pregnant rat spleen is more, causing this damage of the spleen. But in a normal rat, the Salmonella load is reduced or checked to a restricted area and the damage is less. Now, why in a pregnant rat? Because it is obvious that in a pregnant rat, the sex hormone production increases, and with the increase in female sex hormone, naturally the dreadful disease of Salmonella infection increases. Now coming to a very important aspect, where the bacteria is causing gradual modification of the steroid and other hormones. Here we can see the diosgenin is converted to different other progestin or corticosteroids. Now this conversion is brought about in nature by different bacteria. So bacteria like sometimes Bacillus, sometimes it may be Pseudomonas, they convert the different steroid molecule to a series of other sex hormone which can be utilized by the animals growing in the vicinity. So that means that bacteria not only produce hormone but also can result in the conversion and degradation of certain steroid hormone to different other intermediates which can be utilized by other organisms. This is a unique gram-negative Pseudomonas species. It is a specific strain commonly called Chol-1. Now why it is so called? Because it has the unique ability of degrading cholet or bile, and this is how cholesterol is degraded to chenodeoxycholic acid. So cholesterol degradation of this bacteria means a great thing in a person where the cholesterol level is high. If there is infection of this particular Pseudomonas strain, then the cholesterol level will come down in that individual without taking any anti-cholesterol drug. There may be plant sterols produced. These include the cytosterol, the campesterol, the brassicasterol. All these steroid compounds are converted to intermediates, and this sterol degradation in plant also actually tell upon their defense reaction. So a bacteria which is converting plant steroid can make the plant more susceptible to other fungal infection. Now we come to another group of bacteria which is not true bacteria, but it is actually the group of blue-green algae, which is commonly the Cyanobacteria. Cyanobacteria can be unicellular, can be filamentous. In this particular case, there is a unique phoridium. This phoridium is a filamentous cyanobacteria, where we see that the increase in cytokinin level will increase the apical growth of this filament of phoridium. And this particular cyanobacterial cytokinin can actually include different molecules like the kinetin, like the zeatin, or like the simple derivatives of adenine. So adenine in this particular bacteria is converted to different other molecule, which ultimately will bring about their apical growth, and thereby it will pollute the water body, causing water bloom. There is another unique group of cyanobacteria which is called Crucocydopsis, and this particular Crucocydopsis is producing indole acetic acid, which is common auxin. And with the increase in auxin level, their cell divisional rate increase. And what happens is, there is a common barrier within which the cell divides faster and grows faster and colonizes faster. And this is the typical indole butyric acid which is produced by this group of Cyanocystis or Crucocydopsis. In Cyanocystis, what happens is, the Cyanocystis, they are simple rod-like cells which produces different hormone. But this hormone production increases with the presence of some unique molecule which are commonly called anesthetizing molecule. These anesthetizing molecules include the lidocaine and the articaine. And this lidocaine and articaine commonly, they are causing the neuroinhibition in man and in higher mammals. But the presence of this lidocaine in case of the cyanobacteria will induce the production of indole acetic acid, will induce the production of 2,4-D, will induce the production of kinetin, as a result of which their growth increases and their colonization level increases. There is a unique group of cyanobacteria like this filamentous oscillatoria. This oscillatoria, the clearly blue-green color is because of the chlorophyll and because of the C-phycocyanin, which is typically blue in color. And this filamentous cyanobacteria is colonizing in a plant body and will help in the synthesis of different flavonoids or phenylpropanoid compound. And these phenylpropanoids like coumarins, like the chalcones, like the genisteins, these compounds, they help in better living of that plant, increases the antioxidant level, as a result of which the reactive oxygen species come down, and also because of the increase of this flavonoid compound, their nutritional derivatives increase, so they are more nutritious. These plants become more nutritious for mammals, and at the same time they will be protected from different other infection. Now, different crop plants like rice plant, they require sometimes the colonization of cyanobacteria. This colonization either can be directly or it can be through some pteridophyte. Now, like you know, azolla is having the unique cyanobacteria within, that is the Anabaena azole, which is fixing nitrogen, increasing the fertility of the soil. But what we see here is the presence of a unique cyanobacteria called the cytonema inside the internode of a rice plant. And whenever this infection occurs, the length of the internode increases. So in one way, it is promoting the production of a hormone called gibberellin. And this gibberellin in the form of GA9 or GA12 or GA3, these compounds have been isolated in those rice plants affected with cytonema infection. So what does it prove? That because of this cytonema infection, the plant is showing greater apical growth. So thereby we can say that different hormones are being produced by the microbial organisms, including bacteria and cyanobacteria. These hormones may have a definite role in the growth and cell division of this bacteria. At the same time, bacterial infection is determined by the hormone concentration present in human and other animals. At the same time, bacteria has the ability to break down complex steroid or other hormones to relatively simpler compounds. Similarly, cyanobacteria independently can produce different hormones for their growth, development and division. They can colonize in the root or in the shoot of higher crop plants and will produce hormone which will ultimately help in the growth and development of the crop plants.